After completing this learning module, students should be able to list the 5 classes of proteins that function as tumor suppressors, explain hereditary retinoblastoma and how it differs from sporadic retinoblastoma, describe the structure and function of p53, explain why one mutant p53 allele is sufficient to inactivate all p53 function, explain how MDM2 regulates p53 function, explain the genetic defects underlying Lee-Fermini syndrome, explain how defects in DNA repair contribute to cancers, explain why telomerase is upregulated in cancer cells, list evidence supporting the multi-hit hypothesis of cancer, and detail the sequence of mutations that typify progression of colon cancer. Genes in which loss-of-function mutations contribute to transformation are classified as tumor suppressor genes. There are 5 categories of tumor suppressor genes: intracellular proteins that regulate or inhibit progression of a specific stage of the cell cycle, like retinoblastoma protein or the cyclin-dependent kinase inhibitor proteins such as P21 or P16. Receptors for secreted hormones that function to keep cell proliferation in check, such as the TGF-beta receptors. Checkpoint control proteins that arrest the cell cycle if DNA damage or abnormal chromosomes are present, example in P53. Proteins that promote apoptosis when DNA damage or chromosome damage is present in the cell, like the caspases or BAD protein, and enzymes involved in DNA repair. Loss-of-function mutations in DNA repair enzymes greatly increase the rate at which cells accumulate mutations. With rare exception, loss-of-function mutations in tumor suppressor genes act recessively. This means that both alleles of a tumor suppressor gene have to be inactivated before the cell loses tumor suppressor function from that gene. The first tumor suppressor gene that was identified was in patients with inherited retinoblastomas. Retinoblastoma is a rare childhood tumor of the eye. Most cases are sporadic, occurring unilaterally or only in one eye, and present in children between the ages of 1 and 5. The remaining 30 to 40% of patients have a hereditary form of retinoblastoma. These individuals have inherited a germline cancer predisposing mutation in the RB gene on chromosome 13. This usually results in tumors forming in both eyes at a very young age. A developing retina contains about 4 million retinoblasts, or precursor cells, of which occasionally one cell will have an inactivating mutation occur in one retinoblastoma allele. However, tumors will not develop because the other allele is functioning and producing enough retinoblastoma protein to retain control of the cell cycle. However, if that occasional cell that has inactivated one retinoblastoma allele undergoes another loss-of-function mutation in the other retinoblastoma allele, a tumor will develop. This occurs during sporadic retinoblastoma. This is a very rare event and only occurs usually in one eye. Persons that have inherited a germline mutation in one retinoblastoma allele have 4 million retinoblast cells in each retina that already have only one functional retinoblastoma allele. Therefore, spontaneous inactivating mutations in a retinoblastoma allele in any one of those 4 million retinoblasts results in a tumor. In other words, these persons are born with the deck stacked against them. P53 is sometimes referred to as the policeman of the genome. It is a key tumor suppressor protein that halts cell cycle progression when damaged DNA or chromosome abnormalities are detected in the cell. Loss-of-function mutations in p53 are common in many types of cancers. Loss of p53 function means that the cell will now accumulate mutations at a higher rate, which will accelerate its progression to a malignant phenotype. The expression of p53 in the cell is normally very low. Detection of DNA damage triggers a mechanism that results in phosphorylation of p53, preventing its degradation, which allows it to accumulate to critical levels and function as a transcription factor. p53 then induces the expression of cyclin-dependent kinase inhibitor proteins, including p21-CYP, which binds to cyclin CDK complexes and prevents them from functioning. DNA repair mechanisms can then attempt to repair the DNA damage or chromosomal damage so that replication of the cell can continue, or if the DNA damage is so extensive, p53 will induce the expression of pro-apoptotic genes and the cell will undergo programmed cell death. Unlike almost all other tumor suppressor genes, in which loss-of-function mutation in one tumor suppressor allele has no phenotypic effect, a loss-of-function mutation in one p53 allele essentially stops all p53 function in the cell. The oncogenic p53 mutations act as dominant negatives. This is because the wild-type p53 functions as a homotetramer of 4 identical p53 subunits. The tetrameric p53 is assembled from subunits made from both the normal allele and the mutated allele. Any combination of 4 subunits that includes a mutated p53 subunit has diminished activity as a transcription factor, and thus the p53 function is lost in that tetramer. As this figure illustrates, 15 out of 16 homotetramers of p53 would have diminished function. Benzopyrene is a carcinogen found in tobacco smoke. Metabolic activation in epithelial cells in the lung form a potent mutagen that induces G to T transversions. When epithelial cells in culture are treated with benzopyrene, mutations accumulate in p53 at the same positions that are identified in p53 isolated from lung tumors. Here, the length of each bar represents the frequency of a mutation at that location in p53. What becomes clearly obvious is the great majority of these mutations affect its DNA binding domain. This suggests that the effect of mutating p53 on tumorigenesis is due to its ability to bind to DNA and alter gene expression. MDM2, or murine double minute clone 2, is the protein responsible for inducing degradation of p53 in the absence of DNA damage signaling. MDM2 binds to the transactivation domain in the N-terminus of p53, preventing p53 from activating transcription. This allows E3 ubiquitin ligase to ubiquitinolate p53 and target it for degradation in a proteasome. This mechanism maintains p53 at nearly undetectable levels in normal cells. In cells with damaged DNA, checkpoint kinases phosphorylate the N-terminal domain of p53, interfering with its ability to bind to MDM2. This halts the degradation of p53, allowing it to accumulate and activate transcription of genes that will halt the cell cycle. p53 will also activate the expression of the MDM2 gene so that there is plenty of MDM2 protein available so that p53 can be rapidly degraded when the DNA damage has been repaired. The MDM2 gene is amplified in human tumors that maintain normal or wild-type p53. This leads to a loss of p53 function. Therefore, MDM2 is a proto-oncogene, and when overexpressed, is an oncoprotein. Inherited germline mutations in tumor suppressor genes result in a hereditary predisposition for some cancers. Heterozygotes inherit one mutant allele and one wild-type tumor suppressor allele. Since most mutations in tumor suppressor genes act recessively, a somatic mutation of the second allele is necessary to facilitate tumor progression. This is known as loss of heterozygosity. Only one mutation event is needed in one cell, not two in the same cell as required for individuals who are homozygous wild-type for the tumor suppressor phenotype. Therefore, in these individuals, cancers usually occur earlier in life and are often marked by formation of multiple primary tumors. Examples of hereditary mutations in tumor suppressor genes include hereditary retinoblastoma, in which a defect on chromosome 13 inactivates one retinoblastoma allele. Lee-Fermini syndrome is caused by autosomal dominant mutations in a p53 allele. Heterozygotes have a 25-fold greater chance of developing a cancer by age 50 than persons in the normal population. Individuals who inherit a defective allele of the APC gene, or the adenomatosis polyposis coli allele, have a very high probability of developing familial adenoma polyposis. They will develop hundreds of polyps in their colon by their 20s, some of which will go on to become malignant transformations. A great majority of non-inherited or non-familial colorectal cancers also show loss-of-function mutations in the APC gene. Neurofibromatosis type 1 is caused by inactivating mutations in the NF1 gene, a gap protein that regulates RAS activity. Individuals who inherit a defective NF1 allele will develop numerous neurofibromas, presumably as a result of somatic inactivation of the other NF1 allele. Some of these neurofibromas may undergo malignant transformation into neurofibrosarcomas. Hereditary defects in DNA repair mechanisms increase the probability of accumulating mutations in a cell that can be transforming. There are several well-known human hereditary disorders that are associated with DNA repair defects, and all of them have increased cancer susceptibility, including ataxia telangiectasia that leads to susceptibility to lymphomas. It's caused by a mutation in the ataxia telangiectasia mutated gene, which is a kinase that is activated by double-strand DNA breaks. Bloom syndrome, which increases the probability for carcinomas, leukemias, and lymphomas, is caused by a defect in a DNA helicase enzyme. Cockney's syndrome results from mutations in enzymes involved in transcription-coupled repair mechanisms. Fanconi's anemia can be caused by mutation in 15 different genes related to DNA repair, including the BRCA2 gene associated with breast cancers. Hereditary non-polyposis colorectal cancer is caused by defects in the mismatch repair mechanism, and xeroderma pigmentosum, which is caused by defects in the nucleotide excision repair mechanism. Most somatic cells lack telomerase activity, so they have a limited replicative capacity. Every time they divide, their telomeric ends shorten. This means that once they reach a critical level, to avoid unwanted damage to the ends of chromosomes, cells will undergo senescence and stop dividing. If they fail to undergo senescence and continue to replicate, the ends of the chromosomes will become damaged and DNA repair mechanisms will attempt to repair the damage and create end-to-end fusions of unprotected sister chromatid ends. When the cell attempts to separate the sister chromatids, more breaks ensue, resulting in what's known as genomic instability. Eventually, the genomic instability will reach a point at which the cell is no longer viable. To avoid this catastrophic fate, cancer cells will often reactivate telomerase expression to stabilize the genome before this catastrophic crisis event occurs. Loss of p53 function contributes to genomic instability, since damaged telomeric ends would trigger p53 activation and halt cell cycle. Thus, loss of p53 is thought to contribute to genomic instability by promoting telomere shortening upon successive replication, leading to the formation of sister chromatid fusions, which then are broken and then fused and then broken and fused again and again and again as the cells divide. However, in the absence of telomerase activity, this increase in genomic instability adds to the accumulated mutations in a cell, leading to a more malignant phenotype. However, in the absence of telomerase activity, the break-fuse-break cycles would continue until the cells reached a genomic catastrophe point and were no longer viable cells. So many cells upregulate telomerase activity to avoid crossing this threshold level of genomic instability and stabilize the cell's genome with only a certain amount of chromosomal abnormalities. Epidemiological data support the idea that most cancers adhere to the multi-hit hypothesis. That is, a single cell must receive a series of mutational events in order to become malignant or cancerous. This graph illustrates the annual incidence of certain forms of cancers in males by age group. Cancers of the prostate, stomach, skin, rectum, pancreas, and esophagus all show a direct linear relationship between the annual incidence in males and the age at which they are diagnosed. This kind of data supports the idea that it takes years before a cell can accumulate the right types of mutations to become malignant. Experimental evidence from transgenic mice studies also show the combined effects of different mutations on the progression of cancer phenotypes. Transgenic mice overexpressing the oncogene c-MYC develop tumors at a relatively slow rate, as do transgenic mice expressing an oncogenic form of RAS called RAS-D. However, when these mice are crossbred, their offspring, overexpressing MYC and oncogenic RAS, develop tumors at a much higher frequency than the parental lines. Genomic analysis of biopsies taken from colon cancer at various stages of development have identified key steps in a well-ordered series of mutations that leads to malignant cancer. Invariably, the first mutation is a loss of APC, a tumor-suppressing gene. Loss of APC leads to the development of small polyps on the colon wall. These polyps are easily identified by colonoscopies and easily removed and prevent the formation of colon cancers. If the polyp is not removed, a second mutation and activation in the KRAS oncogene leads to the formation of a benign precancerous growth. A subsequent loss-of-function mutation in the DCC gene, or deleted in colon cancer gene, which is a gene involved in SMAD signal transduction, leads to the formation of class II adenoma, a benign growth. A subsequent loss-of-function mutation in the p53 gene leads to a carcinoma, a malignant tumor. Loss of p53 function in these cells leads to a more rapid accumulation of mutations that contribute to the metastatic phenotype of the cancer.